Welcome to Lesson 12.1, Identifying the Substance of Genes. In this lesson, we'll summarize the process of bacterial transformation, describe the role of bacteriophages in identifying genetic material, and identify the role of DNA in heredity. Like many stories in science, the discovery of the chemical nature of the gene began with an investigator who was actually looking for something else. In 1928, the British scientist Frederick Griffith was trying to figure out how bacteria make people sick. More specifically, Griffith wanted to learn how certain types of bacteria produce the serious lung illness known as pneumonia. Griffith had isolated two very similar types of bacteria from mice. These were actually two different varieties or strains of the same bacterial species. Both strains grew very well in culture plates in Griffith's lab, but only one of them caused pneumonia. When Griffith injected mice with disease-causing bacteria, the mice developed pneumonia and died. When he injected mice with harmless bacteria, the mice stayed healthy. Griffith wondered what made the first group of mice get pneumonia. Perhaps the S-strain bacteria produced a toxin to make the mice sick. To find out, he ran a series of experiments. First, Griffith took a culture of the S strain, heated the cells to kill them, then injected the heat-killed bacteria into laboratory mice. The mice survived, suggesting that the cause of pneumonia was not a toxin from these disease-causing bacteria. In Griffith's next experiment, he mixed the heat-killed S-strain bacteria with live, harmless bacteria from the R strain. This mixture he injected into laboratory mice. By themselves, neither type of bacteria should have made the mice sick. To Griffith's surprise, however, the injected mice developed pneumonia and many died. When he examined the lungs of these mice, he found them to be filled not with the harmless bacteria, but with the disease-causing bacteria. How could that happen if the S-strain cells were dead? Somehow the heat-killed bacteria passed their disease-causing ability to the harmless bacteria. Griffith reasoned that when he mixed the two types of bacteria together, some chemical factor transferred from the heat-killed cells of the S strain into the live cells of the R strain. This chemical compound, he hypothesized, must contain information that could change harmless bacteria into disease-causing ones. He called this process transformation, because one type of bacteria had been changed permanently into another. Because the ability to cause disease was inherited by the offspring of the transformed bacteria, Griffith concluded that the transforming factor had to be a gene. Later, a group of scientists led by Canadian biologist Oswald Avery wanted to determine which molecule in the heat-killed bacteria was most important for transformation. They reasoned that if they could find this particular molecule, it might reveal the chemical nature of the gene. Avery and his team extracted a mixture of various molecules from the heat-killed bacteria. They carefully treated this mixture with enzymes that destroyed proteins, lipids, carbohydrates, and some other molecules, including the nucleic acid RNA. Transformation still occurred. Clearly, since those molecules had been destroyed, none of them could have been responsible for transformation. Avery's team repeated the experiment one more time. This time, they used enzymes that would break down a different nucleic acid, DNA. When they destroyed the DNA in the mixture, transformation did not occur. There was just one possible explanation for these results. DNA was the transforming factor. By observing bacterial transformation, Avery and other scientists discovered that the nucleic acid DNA stores and transmits genetic information from one generation of bacteria to the next. The work by Avery and his team was confirmed in the 1950s by a research team that was studying viruses. The virus that was studied was a bacteriophage, a kind of virus that infects bacteria. When a bacteriophage infects a bacterium, it attaches to the surface of the bacteria cell and injects its genetic information into it. The viral genes act to produce many new bacteriophages, which gradually destroy the bacterium. When the cell splits open, hundreds of new viruses burst out. Here's a micrograph showing a bacteriophage, green in color, invading an E. coli bacterium, the gold-colored cell. Alfred Hershey and Martha Chase used different radioactive markers to label the DNA and proteins of bacteriophages. The bacteriophages injected only DNA, not proteins, into bacteria cells. Once the phages infected the bacterium, they noticed radioactivity inside the bacterium that had the radioactive tag in DNA, but there was no radioactivity inside the bacterium with the radioactive tag in the protein coat. This experiment confirmed Avery's results, convincing many scientists that DNA was the genetic material found in genes, not just viruses and bacteria, but in all living cells. DNA has 3 essential roles. We'll learn more about each of these roles in future lessons. DNA is responsible for storing information. It's also responsible for copying that information for future daughter cells. And finally, DNA's role is to help transmit information to daughter cells.